One other thing I want to mention, I'm trying to record all your participations. So since this will be the last actual class meeting that you have opportunity to participate in, I will update the spreadsheet and I'll send it to you later this week. And please take a close look at it and send me an email about any omissions on your record.
So today will be the last day for participation. The last 2 days will have nothing, right?
That's correct. The last two class periods, we will not actually be meeting online. So Wednesday, you have the entire class period is devoted to test 3. And again, you have all day to take that test. And then the following week on Wednesday, the 15th, you have all day to take the final. And again, the class will not meet on either of those days. So today is the last day that we'll have an actual class meeting.
Oh.
You all and me.
Oh, okay. Where's that calendar? Guess I want to see. Let's see.
Well, I'd like to start now with the questions that you've submitted. I'll share my screen.
Wait, one more question. Okay, you said we have the test on the 8th, so we have no class, and then we have no class on the 15th. So what about the 13th? We don't have no class at all. We don't come that Monday.
Yeah, that's right. You do not come on Monday because that's finals week. There are no classes.
Oh, okay, at all. Okay, I just want to make sure. Okay.
So today is the last day that we'll actually have a class meeting.
Okay. Okay.
So getting back to the questions that you submitted, I've tried to consolidate and parse them out so that I can discuss them one at a time. And anything that you might have had in mind that I don't cover, just speak up and let me know. So do you all see, do enzymes have functions?
Yes.
Okay, make that a little bigger. So this is one question that was submitted. Do enzymes have functions other than digestion of food? So I thought a good place to start with this question was to deal with the role of enzymes in the digestion of food. And with respect to enzymes, remember that any word that ends in ACE, A-S-E, is probably an enzyme. And the corollary of that is any word that ends in OSE, O-S-E, is probably a sugar. So how is food digested? Well, you may remember this picture from when we talked about cystic fibrosis. So let's just go through the effects of digestive enzymes. So in your mouth, there are a number of digestive enzymes called salivary enzymes, one of which is amylase, which breaks down starches into disaccharides. Then in the stomach, there are a couple of enzymes. The main one that you should be concerned about is pepsin. Pepsin is in the stomach and it breaks down proteins into large peptides. And the thing to remember about this is that the stomach has a pH of 2. Pepsin has a pH optimum of 2 because it has to function in this very acidic environment. So have any of you thought about why the stomach has such a low pH?
I haven't.
Okay, well, think about what you eat. And what you eat is almost never sterile. It's full of microorganisms. And microorganisms, especially bacteria, cannot stand acid. So the stomach acid kills most of the organisms that you ingest immediately. And after that, the food, partially digested food, goes into the small intestine. First part is called the duodenum. And here the pH changes dramatically from 2 to about 7. So all of the enzymes from here on have pH optima of about 7. And these include amylase, which continues the breakdown of starch. Trypsin, trypsin is another peptidase because it continues the breakdown of protein, but it has a different pH optimum than does pepsin. And lipases break down fats. Also in the small intestine, you have more enzymes for breaking down sugars and peptides. So as the food travels down the small intestine, you have all these different enzymes digesting the different components of food. And one thing to remember about digestion, and that is the breakdown of fat. Fat is not soluble in water, yet all of these enzymes require an aqueous environment. So how do we deal with fat? That's where the gallbladder comes in. It produces bile. And if you remember from cystic fibrosis, bile contains these molecules which are called amphipathic. That means they're negative on one end and positive on the other. And so they're essentially soaps. We talked about how soap cleans skin, cleans anything, because it emulsifies fat. It makes fat soluble in water. So that's a complication of digestion, that fats are a different category. They have to be made soluble in water before lipases can digest them.
So you said the gallbladder, it can break down the fats?
Produces bile, which emulsifies fat. And then enzymes that are in the small intestine, known as lipases, they actually digest the fats, which are long chains of fatty acid, break them down into fatty acids, which then can be used as energy.
So what if the gallbladder is gone? I mean, if you don't have one.
If you don't have one, you got to watch your diet.
Oh.
Or you can take supplements to supplement the lack of bile. But from what I understand, and maybe some of you know more about this, people who have had gallbladder surgery are usually on strict diets.
I ain't, but I had mine removed years ago. That's why I was asking, because they never told me any of that.
Professor, I have a question.
Okay. What's your name? Oh, Gabriela.
Gabriela. Gabriela Galvez. So like me, I have no gallbladder, okay? Like 4 years ago, they took off my gallbladder. So you say that what happened with my fats? I believe that I have a healthy diet. After that, I changed the way that I eat.
Do you
So
have
I
a special diet?
Not a special diet, but I'm taking care about sugar. And you know, as we know, everything has sugar. Like every food, like the rice and cheese. You know, like I have a diet based on protein. Most of what I eat is protein, and my diet is based on protein. But what happened with the fat? Because my problem is, I'm sorry to say that, but I have a constipation problems. I'm in charge of that. I'm fine with that, but I know then I have the problem. So it's probably because I have no the gallbladder and the enzymes and the fats not processing well on my intestine. Could be like that, something like that.
Yeah, if you have no gallbladder, I'm fairly sure you have a problem digesting fats. So I suspect that your diet includes low-fat content, and you may take supplements to help you digest fats.
I don't.
So
Yes, I am.
So this is Steve Stinson. So if she doesn't take those supplements, then would it cause? I know you're not a doctor per se, but would it cause constipation?
It would cause some kind of digestive disturbance. So either constipation or the opposite, diarrhea.
Oh, that's probably why I have that. Okay. I'm Yolanda. I'm the first one that mentioned my gallbladder's been taken away years ago. And they didn't give me no instructions or any of that. And I've had children since then and nothing different. It's just, but I do have the diarrhea thing. I mean, I don't have it. It's just if I eat certain things, then it's, I don't have that constipation.
The prediction would be that if you eat fat, it's not going to be digested. It'll get into your small intestine and eventually into your large intestine undigested. But that would cause a digestive disturbance. So in your case, it sounds like it causes diarrhea.
I thought I thought because I was doing that, I inherited it from my great grandmother because she had that issue, but I don't know if she had her gallbladder or didn't have it. Like I said, she was my great grandmother, and I know like greens, ice cream, stuff like that. So I just put it off as I inherited. I didn't know until now that the gallbladder had something to do with it.
Yeah, it has everything to do with fats.
Oh.
And Professor, I have another question. That the probiotics help with the enzymes and the fat.
Say that again.
Okay, the probiotics will help with the fat and the enzymes. Could help, during the process of digestion process.
Yeah, probiotics are poorly defined dietary supplements. So it depends what's in your probiotic. If your probiotic has some component that helps you emulsify fats, then yes, it would help you in your digestion. So you'd have to check on the label of your probiotic and see what's in it.
Yeah, it helped me in my case, helped me with my problems, and I'm on the probiotics right now, so it's very good. It really helped with me.
Yeah, so that's a dietary supplement, the probiotics, and probiotics usually have live microorganisms in them. So the microorganisms that you're taking in your supplement are probably helping you digest fat.
Thank you.
Okay, so this entire discussion has been about a particular class of enzymes called digestive enzymes. And the question that someone asked is this the only type of enzymes that we have? And the answer is no. There are many other types of enzymes, and now I want to go through them as classes. They can be classed according to what their functions are. So it can be hydrolase, isomerase, lyase, oxidoreductase, synthetases, and transferases. And these do various different things. These are classes. And within each class, there are many different specific enzymes. So remember we talked about catabolic reactions and anabolic reactions. So catabolic reactions tear things apart. And that is, that's how we make polymers. Excuse me, that's how we break down polymers to derive energy from them. So we just talked about lipases and proteases. And hydrolysis, hydro means water. So it means tearing things apart by adding water. And let me just contrast that to anabolic processes, which are catalyzed by what are known as synthetases. These join two molecules together by the formation of a new bond. And this is called dehydration synthesis. So you should remember that when two molecules are joined together, water is taken out, and it's called dehydration synthesis. So when you tear things apart, you add water. When you join them together, you subtract water. Now, another completely different class of enzymes are involved in the metabolism of nucleic acids. We spent a lot of time talking about how DNA replicates itself. And this is a picture of a screenshot that we've gone over before. And it points out the different enzymes that are involved in just one step of DNA replication, the exact copying of DNA from a template molecule to a new molecule. So you have a helicase, a primase, DNA polymerase 3, RNase H, and DNA ligase. Notice that they all end in ASE, and they're all enzymes involved in DNA replication. So that's another class of enzymes, has nothing to do with digestion of food, has everything to do with copying DNA. And there were actually several questions about what's the relationship between enzymes and energy. Well, it turns out that there are many enzymes involved in energy metabolism, in both the capture of energy and the release of energy. So enzymes are required to produce energy and to use energy, and this is involved primarily in the two sections that we covered on photosynthesis on the one hand, and respiration on the other hand, respiration being the biological oxidation of food. So photosynthesis, you will recall, has two basic reactions called the light reaction and the dark reaction. And I have here a diagram of the light reaction. And the light reaction occurs in the chloroplast in this membrane called the thylakoid. So remember that. Thylakoid membrane is where light is received, light from the sun, by these two photosystems, photosystem 1 and photosystem 2. So when a photon of light is received by photosystem 2 or 1, the water is split from H2O, it releases O2, and you end up with a hydrogen atom, which you recall is nothing but a proton and an electron, which is excited, and it goes through this electron, the electron part of it goes through this electron transport chain and ends up providing energy in the form of ATP and NADPH for the dark reaction. And notice that there are enzymes here: cytochrome oxidase, succinate dehydrogenase, NADH dehydrogenase, cytochrome oxidase again over here. So these are the dehydrogenases and the oxidases are what cause the electron transport chain to occur. And for that, this is a good question to synthesize a lot of information. For each electron transport chain, remember, we have biological oxidation and reduction. So if there's an organic molecule that has two extra protons, two extra hydrogen atoms, they're transferred. This would be an oxidized molecule. If it transfers these electrons to a reduced molecule, it now becomes oxidized and the other molecule becomes reduced. And this is like a toggle switch. It goes back and forth from oxidized to reduced, hand off to another molecule, reduced to oxidized, hand off to another molecule, and it just keeps going through the thylakoid membrane. And finally, you end up with water being produced. The point here is that each of these handoffs from one electron carrier to another is catalyzed by an enzyme. And it's not the same enzyme. These reactions do not occur spontaneously. Each redox reaction requires a specific enzyme to catalyze it. And that's why you have names like NADPH dehydrogenase, cytochrome oxidase. All of these steps along the way, each requires a separate enzyme. And so this is the light reaction, which produces ATP and NADPH, which within the chloroplast, here's the chloroplast, go to the dark reaction, also known as the Calvin cycle. So the light reactions produce ATP and NADPH, go to the Calvin cycle, which takes carbon dioxide, a gas, from the air and fixes it into a solid compound. And this amazing enzyme that does that is called rubisco. So rubisco is the first step in the Calvin cycle. Then there are a whole series of intermediate steps. Each of these steps is catalyzed by a different enzyme. So you remember that the Calvin cycle uses ATP, it uses NADPH to generate glucose. So we have the overall reaction from the light reaction and the dark reaction. You go from water plus sunlight to carbon dioxide and sugar, usually glucose. And that's the end of energy production, photosynthesis. It has a light reaction and a dark reaction, and each of them involves a lot of different enzymes. Now, if we go from photosynthesis to respiration, we start with glycolysis, start with glucose, because that's the product of photosynthesis. So now we've captured energy from the sun in photosynthesis. Now we need to derive energy from that. So we start with glucose, which is the result of the Calvin cycle. And we have a process called glycolysis, and it's usually shown like this, where glucose is split into 2 molecules of pyruvate. Glucose is a 6-carbon sugar. Pyruvate is a 3-carbon compound. And so you get a small amount of ATP by doing this, a small amount of NADH, and you get 2 molecules of pyruvate. However, glycolysis is not a 1-step reaction, it's a 9-step reaction from glucose over to pyruvate. And each step is catalyzed by a different enzyme. So these are all enzymes that have nothing to do with food digestion. They're entirely involved in deriving energy from digested food. Any questions? Are you following what I'm describing here? So we go from pyruvate. Pyruvate goes to acetyl-CoA, which is the first step in the Krebs cycle. The Krebs cycle occurs only in the mitochondrion, and this is where we derive energy. This is where oxidative phosphorylation comes from. So the Krebs cycle yields carbon dioxide. It yields reducing power in the form of NADH and FADH2, and a little bit of ATP at substrate level phosphorylation. So the main purpose of the Krebs cycle is to produce reducing power, electrons and protons. So they go spinning off as the Krebs cycle turns. And here you have where the action actually occurs. Within the cell, you have mitochondria. And within the mitochondria, we have oxidative phosphorylation, starting with glucose converted to pyruvate in the cytoplasm. Pyruvate crosses the membrane and goes to acetyl-CoA, which enters the Krebs cycle, TCA cycle, which produces a little bit of ATP and a lot of NADH and FADH2. So what happens with these, this reducing power? Well, the electrons go into the mitochondrial membrane, the inner mitochondrial membrane. So electrons travel through this membrane through an electron transport chain, which is very similar to the electron transport chain that we see in photosynthesis, in the light reaction of photosynthesis. This occurs in the thylakoid membrane. In the mitochondrion, it's the inner mitochondrial membrane where this electron transport chain is occurring. And it results in, there's a whole series of redox reactions. And at the end, oxygen is the final electron acceptor that accepts an electron and makes water. But while the electrons are passing through the membrane, they're depositing protons in this intermembrane space. These protons, at some point, are transferred back out of the membrane by a process known as chemiosmosis. That's these little purple and blue structures. And each of these, by passing protons through, generates ATP from ADP. Now, this is oxidative phosphorylation, and that's where we get most of the energy that we use. But again, we're talking about enzymes, different types of enzymes. And again, there's a whole series of enzymes involved in glycolysis, another series in the Krebs cycle. And then once the reducing power is generated in the Krebs cycle, another whole series of enzymes for the electron transport chain, and then another enzyme complex for chemiosmosis that generates ATP. So I hope I've left you with the knowledge that there are a lot of enzymes. Nothing happens in a cell that's not catalyzed with an enzyme. So in addition to digestion, we have nucleic acid metabolism, and we have a huge number of enzymes involved in the production of energy and the utilization of energy. So, but not all proteins are enzymes. The converse is all enzymes are proteins, but not all proteins are enzymes. So we've been talking entirely about enzymes here, but remember, when we talked about biomolecules, we talked about types of proteins, and enzymes is a class here, clear at the end of the list. So there are structural, contractile, storage proteins, protective, transport signal, communication, and toxins. These are all proteins, they're not enzymes, and they each have a specific function. But you see the distinction I'm making here between the various different classes of enzymes and classes of proteins. All right, change topics. There's another question or several questions about evolution. And one question was, what determines when it will occur? So what causes evolution to occur? It occurs because of genetic changes. And these genetic changes can be mutations. They can derive from meiosis, where you have crossing over and independent assortment, which creates genetic variation. And then we have variation generated by fertilization of eggs with sperm. And this occurs in both plants and animals. So we have this constant generation of new genotypes, new sets of genes. That's where natural selection comes in, because it can sort through all this new genetic material and identify the organisms that are best at reproducing. So the ones that produce the most offspring are the ones that evolve. So what determines when evolution occurs? Two things. One is genetic change and natural selection, and also the creation of new ecological niches. So when the climate changes, for example, and a new ecological niche is produced, now natural selection can sort through the genotypes that genetic change has produced and identify which ones can reproduce best in this new niche. And now we have the generation of a new species. And I'm reminding you of this little diagram of giraffes, which illustrates how it happens. So among an ancestral population of giraffes, some had longer necks than others. So both types and everything in between, they all reproduced. But it turned out that those with longer necks produced more offspring, more baby giraffes, than those with short necks. So the ones with long necks over generations outcompeted those with short necks, so that finally you end up with a population that has nothing but giraffes with long necks. So genetic change is not inherited, it's selected for.
Will we have to describe a diagram like this on a test by Darwin?
I might ask you a question about this, but I won't ask you to draw pictures of it.
And explain it? Okay.
Then there were a couple of questions about human evolution and the involvement of humans with Neanderthals. So one question was, if both humans and Neanderthals were from Africa, why didn't they mate in Africa? Remember, they mated in Europe, Northern Europe. So the short answer to that is that these two species were in Africa at different times. They didn't actually interact with each other in Africa because they weren't there at the same time. So I remind you, I show you again this picture of the migrations of humans and of Neanderthals. All humans and species related to humans evolved in Africa. Then Neanderthals migrated out. Now, Neanderthals, let me go to this next diagram here. Neanderthals are much older than humans. So they came into existence about 600,000 years ago. Humans didn't arrive until about 200,000 years ago, so 400,000 years later. Now, before humans evolved, Neanderthals migrated out of Africa into Eurasia, Europe and Asia. Then their lineage in Africa became extinct. But humans derived from the same lineage, and they migrated out of Africa about 65,000 years ago, much later than Neanderthals did. But they overlapped with Neanderthals in Eurasia between 47 and 65,000 years ago. And this is when they mated. But another, I think, point of confusion is that humans have a lot of relatives, close relatives, all of which are now extinct, but they all derive from one ancestor down here called a hominin ancestor. And that ancestor gave rise to chimpanzees, which are the closest ape relatives that we have. And then this whole series of what you could think of as cousins that all arose and evolved in Africa and all became extinct in Africa. And finally, we end up here with Homo sapiens, that's us, and Neanderthals, who actually interacted sexually in Europe. I found this picture, been looking at it, trying to decide which one is the Neanderthal and which one is the human. This is somebody's imagination based on DNA sequences. So getting back to the answer to the question, why didn't they mate in Africa? It's because the Neanderthals evolved much earlier than humans. They migrated out and then went extinct in Africa. Humans evolved later, and then they migrated out and overlapped there was about a 40,000 year overlap between when humans arrived and when Neanderthals became extinct. So if you read here, Neanderthals left Africa 400,000 years ago. Humans didn't leave until 60,000 years ago. So 340,000 years after Neanderthals. Neanderthals used all this time to acquire mutations to adapt to the harsh environment. Humans took a shortcut by mating with Neanderthals and acquiring some of their genes, which we still have. All ethnic groups derived from Eurasia have Neanderthal DNA in them. Now, I just ran across something interesting, just new information just from this year, that there is a small amount of Neanderthal DNA in the African population. And this is not due to mating in Africa. It's due to modern humans migrating back from Eurasia back to Africa. So now they've acquired Neanderthal DNA, they've migrated back to Africa, and now by mating with Africans can introduce small amounts of Neanderthal DNA to African populations. So if you just read this, European ancestors introduced a substantial part of Neanderthal sequence to Africa, and it spread throughout the continent. And they have evidence that migrations both out of Africa and then back into Africa have played a prominent role in shaping patterns of human genetic variation. So any questions about Neanderthals and humans, where they overlap, where they met, when they evolved? So another question about climate. What's the explanation for the lack of cold weather in 2021 as compared to previous years? Is it due to pollution in the air? The answer to that is yes, it is due to pollution, and the main pollutant is carbon dioxide. But just to review, let's think about greenhouse effect and what causes it. The greenhouse effect causes warming of the entire planet. The sunlight strikes the earth. Some of its energy is absorbed and some is reflected back out into space. What carbon dioxide and a few other gases do is form a layer in the upper atmosphere of the earth, which blocks some of this reflected sunlight. And that has the same effect as sun shining into a greenhouse. That is, the heat cannot get out. And so the planet is warming. And the more carbon dioxide we produce, the more the planet will warm. So you have a whole slide set on climate change, which elaborates on carbon dioxide and what we can do about it. But the question here is why the lack of cold weather in 2021? And it raises this issue of the difference between weather and climate. So many people are unclear about this. They think of weather as the same thing as climate, and in fact, it's not. It's very different. So weather is the atmosphere at a point in time and at a particular place. So in San Diego today, the atmospheric conditions are the weather in San Diego. Climate, on the other hand, is weather averaged over a long period, usually 30 years. So you can see that in these graphs that there's a lot of zigzagging up and down over the short term, but over the long term, there are trends. Well, the short term is the weather at a point in time. The long term is the climate.
Professor.
Yeah.
When it comes to climate change, because I paid attention to when they went from global warming to started calling it climate change. And I realize that it's a long period, but since they have no actual documentation of, let's say, 200 years ago, how do they make those determinations that we haven't cycled? Well, let's just say 1,000 years ago, we didn't cycle through and actually have similarities to the way that climate is now.
Yeah, that's a good point. And the answer is speed. So if you just look at this one graph, or go back to this graph, thousands of years, so 800,000 years ago, this is looking at carbon dioxide concentration, 800,000 years ago. That is a long time. And you can see that the carbon dioxide concentration has varied a lot over these many hundreds of thousands of years. But these changes have been slow. And so the climate, the Earth's climate has changed, but it's changed slowly. And when it has changed rapidly, like in the case of the extinction of the dinosaurs, there was a catastrophic event. Something came from outer space, landed on Earth, and caused a huge disruption of the climate and caused mass extinctions. That's because it happened over a short period of time. Over a long period of time, life forms can adapt to climate changes. So the problem we have now is that we humans are causing the climate to change very fast. And species that are here are becoming extinct much more rapidly than they would naturally. Make sense, Steve?
Sure.
There's a difference between historic climate records and current ones is the speed with which the changes are happening. There was a request by someone and I think I interpreted this right, ask to go over again how you analyze DNA sequence and how you make the relationship between DNA sequence and peptide sequence. We've gone over this and we've had it on tests, and I'm fairly sure we're going to have another problem like this on an upcoming test, either test 3 or the final. So let me just review it so that you'll have no problem if you see this something like it on a test. So what I would do is give you a sequence of a piece of DNA. So that sequence is here. And then I'll give you a mutation. So this T mutates to an A. And then I would ask you, what is the effect on the peptide? Well, the way you approach this is first start with this original strand of DNA and transcribe it to messenger RNA. And then you translate that messenger RNA into protein using a codon table, like you see here. And if any question like this, I would provide you with a codon table. So in class, I gave you two examples, mutation 1 and mutation 2. So here, the T becomes an A, and in the next mutation, the G becomes an A. And in both cases, what is the effect on the peptide? Well, here's a summary of how you do that. In the case of wild type, you take the DNA, translate it to RNA. Remember that every A, when it's translated to RNA, is a U. So this would be the sequence after transcription. So then you look up each of these codons in a codon table like this. And we see that AUG is met, UCA is serine, AAU is asparagine, and so on. And UGA is a stop codon. And there is no more synthesis of peptide after that. If we look at what happens with the first mutation, where A is mutated now to U, we start out with AUG, which is methionine. Again, we're going from the codon usage table. And so now in the messenger RNA, the codon is UCU, not UCA, UCU. And so if we look at the codon usage table, UCU and UCA are both serine. So the sequence of the peptide does not change with mutant 1. And so because there is a mutation, but it doesn't change the peptide sequence, it's called a silent mutation. Now, if we look at mutant 2, this G here in the original sequence becomes an A. And so when that's transcribed, the codon is now, instead of UCU, it's instead of UCA, it's UUA. So now if we look in the codon table, UUA is leucine. So now the sequence, instead of being met serine asparagine, it's met leucine asparagine. And this is called a missense mutation. And I've got here the descriptions of the definitions of the different types of mutations. A silent mutation has no effect. A missense mutation is the result of one base pair change, which alters the protein. Then keep in mind that there are other types, like a frame shift, which is either the addition or the loss of a single base pair. And it alters the reading frame so that you get an entirely new sequence of peptide. And then there's a nonsense mutation, which is the introduction, a base pair change, which introduces a stop codon. So the ribosome stops translating whenever it comes to a stop codon, and the peptide is truncated, it's shortened. So review this for yourselves so that you'll be able to do any question I might ask about this.
Excuse me, I have a question. So we have to stop when it says stop. We have to stop even though there is more like to translate, or because it's as said, it's not, doesn't work anymore.
Right. Stop is a hard stop. There's no more ribosomal activity. Once the ribosome hits a stop codon, everything falls apart.
Yeah, I got a quick question too.
Mhm.
I remember on the test, I wrote down stop and it was actually wrong. And are we, when we write it down on the test, are we supposed to just give what the, what am I saying? Give the peptide chain without saying stop in it, or?
Oh, you can put the stop in or not.
Okay.
I review every answer anyway, so I can override the speed grader if the speed grader is wrong. So you can either put stop in or not, but don't translate beyond stop.
Okay, gotcha.
Anything else? Okay, another question. This is a very good question. Why do nucleases have site specificity and the other depolymerases don't? So I got to adjust your mind now to different types of enzymes that digest polymers. You eat food, and food is composed almost entirely of polymers, carbohydrates, fats, proteins, and nucleic acids. So the enzymes that break down carbohydrates into sugars are called carbohydrases, like cellulase and amylase. Fats are broken down by lipases. Proteins are broken down by proteases, such as pepsin and trypsin. And nucleic acids are broken down by nucleases, DNase and RNase. The question is, why are the nucleases different? They can be different. So let me skip down to this illustration. Proteases digest proteins, and they can do that by either nibbling off the ends. Some are specific for the N terminal, some for the carboxyl terminal, and some cut in the middle, but all the digestion is non-specific. You start with the protein, you end up with amino acids. Similarly with lipases, you start with the fat, you end up with fatty acids. Same with carbohydrates. You start with these long sugar chains, and carbohydrates can nibble at the ends, chop in the middle, but it's all non-specific. You just break the entire chain down into monomer sugars. Now, not so with nucleases. Nucleases can be specific, and if you remember, nucleases such as BAM-H1, EcoR1, HinD3, they only cut at specific palindromes, which are usually 6 nucleotides long. And so if you take a stretch of DNA, you can see that BAM-H1 cuts at certain sites. EcoR1 cuts at certain sites, and they're different between BAM-H1 and EcoR1. HinD3 is yet different from Eco and BAM. So each of them has these very specific sites at which they cut. And we humans find this very valuable because it allows us to do genetic engineering. But bacteria don't have that goal at all. So the site-specific nucleases protect bacteria from invading viruses. Viruses are composed primarily of nucleic acids. So the bacteria don't care about proteases, lipase, and carbohydrases. They care about nucleases because they would like to degrade the nucleic acid of the invading viruses. And so why do they need site-specific nucleases? It's to protect, for them to distinguish their own DNA from the alien DNA, the attacking virus. And so you can think of this, remember we talked about forensics and how a DNA analysis is used to identify individuals, which suspect did it. Well, bacteria have done the same thing by recognizing their own the restriction enzyme sites in their own DNA. They can distinguish that from the restriction enzyme sites in viral DNA. And so the ones they don't recognize, they cut, and that destroys the virus. So it's like a sort of immune system that bacteria have to protect themselves from viruses. And that's why it's necessary for them to have nucleases that are site-specific. They didn't do it as a favor to humans. They did it to protect themselves. And as a result, humans have been able to take advantage of this for genetic engineering. So does that make sense? Why nucleases have site specificity and nobody else does? It's a very good point to make. Someone asked about gene therapy. Just a couple of points to make about this. In this diagram, you could do gene therapy either by adding a gene or replacing or repairing a gene. So in the case of gene addition, you have a mutant here with the red dot, and you have a wild type without the red dot. If you add the wild type gene, it now can override the mutant copy. The other way to do it is to replace the mutant copy with a wild type copy. And that's called gene repair. And now we have CRISPR. And just to remind you, CRISPR has the merits of being site-specific. That is, you can target the point of DNA you want to mutate precisely, and it's universal. It works in any organism. Those are the two things you need to remember about CRISPR. And the way it works is you start out by making a guide RNA. This guide RNA is complementary to the target, to what you want to mutate. And you introduce that along with this enzyme known as Cas9. Cas9 is a non-specific nuclease. It will, in principle, cut anywhere, but it's constrained to cut only where the guide RNA targets. So the guide RNA directs a cut to be made at a particular, very precise point in the DNA sequence. And it's then that mutation which can be manipulated for whatever the geneticist wants. So one thing that came up in our discussion when we talked about this is, can you edit the human germline? If you do editing in somatic cells, you're only changing cells that are already exist in your body. And if you have offspring, that they will not transmit the edited DNA. However, if you edit the germline, then you've changed the organism genetically. So you can imagine how this might be done. If you had an egg in a petri dish and sperm from a donor, a single sperm would fertilize that egg. If you added to this mix CRISPR, so Cas9 and the guide RNA, now you can make the mutations in the embryo. The embryo then would be transplanted into a human female to the uterus, where it would develop into a baby and eventually be born. And now it will be born with whatever genetic change CRISPR had made. And this it is not done with humans. It can be done, and it is done with other animals, not with humans. There was a related question about how would natural selection and evolution affect us in the future. Are we going to have more physical changes? Well, because of CRISPR and gene editing technology, humans can now control the characteristics of any species, including our own species. So for example, things like disease susceptibility, relative intelligence or athletic ability, on and on. You can think of a lot of different phenotypes that could be manipulated using gene editing. So that's point one. Point two is ethical issues. So scientists can deliver technology to make these genetic changes, but society has to deal with the ethics. What it would be good to do and what would be bad for the species. And the third point here about natural selection and long-term evolution of our species in the future. The long-term prognosis is that this planet, like all planets, will eventually be uninhabitable. And if humans are to continue to exist, they will have to go somewhere else, colonize space some way. And I pointed out when we talked about this earlier that there's this new book out, published just this year. The title of the book is The Next 500 Years. And this author deals with all these issues, and he actually lays out phases, I think 9 different phases of advancement of humans, starting with genome editing and going on to space colonization. And then as an example of how humans or any animal might be made more adaptable to a changing climate, I took the example of radiation resistance. And these little organisms called tardigrades, which are highly resistant to extreme environments. They're resistant to radiation, high and low temperatures, pressures, lack of oxygen. They can go without water, and they don't need to eat for long periods of time. So all of these characteristics are controlled genetically. So to show how this could actually work, there was a lab that identified a gene for radiation resistance. They cloned that gene from the tardigrade introduced it into human cells. Now, these are human cells in culture. They're not in a human body, and made these cells highly resistant to X-rays. So it's an example of how you can take a trait like radiation resistance from one organism and transfer that gene and the phenotype to a completely different organism, including humans. That's the end of the questions that you submitted. And we're at the end of the class period, but if you have any additional questions or comments, speak up.
I just want to say it's been very interesting for the class. So I don't see any talk to any of you guys again. It was nice meeting all of you guys. Have happy holidays. No drinking and driving. Stay safe. And maybe I'll see you in another class.
Yeah, well, I'd like to see you and everyone again sometime. I don't know if that will happen, but I would like to say that I very much enjoyed this class. You've been one of the best classes that I have had. Responsive and thoughtful and provocative and supportive. We've had some snags as we've gone along, and you have all been very supportive of solving the problems as they have arisen. So I appreciate that, and I thank you all.
Okay. Bye-bye, guys. This is it.
Thank you for everything. Thank you.
Okay, well,
Professor, I have a question real quick, though.
sure.
Are you doing lab?
Am I doing a lab?
Yes.
No, there is a lab offered in this course, but I don't teach it.
Okay, I know I was trying to sign up for a lab, and the counselor said they was trying to get the same person that I took the lecture from to take the lab as well, and I didn't see your name, so.
Yeah,
Okay.
well, I'm not involved in labs.
Okay, all right.
Wish I was, but I'm not needed for that.
All right. All right. Okay, bye, guys.
Bye, y'all. Have a good one. Have a good
You
holidays.
too. Okay, you
Bye.
too.
Good luck with the test. Good luck with the
Bye-bye.
test, everybody. Thank you,
Yeah,
Yeah, you
Professor.
too.
I say the same thing. Good luck with the test.
Thank you.
Thank you so much,
Okay,
Professor.
bye.
Have a happy
Professor.
holidays.
Yeah,
Is today the last day or tomorrow?
today's the last day that the class meets. So this is the
Oh,
last
okay.
time I'll see you in Bio 130. Hopefully I might see you at some other venue in the future, but not in Bio 130.
Oh, okay. I've got my days totally mixed up.
Well, I better fix that because
So Wednesday is the final?
no, Wednesday is test 3. So
Right, that's what I thought. Okay. Whew.
And then
Oh.
a week from Wednesday, December 15th will be the final.
Okay. So we're not meeting on the 1st. I mean, on the Monday.
That's right. The next week is finals week. So officially, this week is the last week of classes. Next week,
Okay,
there are no
so-
classes, just tests.
Well, I have a question for you before you go. I know we're going long. I wanted to be able to use the information from your recordings for future references. Can we record them and have them so that we can have that information, or is once the class is over, it's just gone?
No, as long as you have access to the recordings, you can use them. I don't know how long well, they're all YouTube videos. So you should be able to get them from YouTube for as long as YouTube retains them.
Okay, cool. So is there a YouTube address for them?
For all of them, there's a YouTube address for each one individually.
Okay,
So you have all those addresses. You might want to collect them into a single document so that you, and also indicate what topic is on each of those addresses.
Okay. All right. Thank you so much. Have a great rest of the year.
Okay, you too. And
All right, bye.
as I say, it's been a pleasure working with you.
All right. Bye.
Okay,
Likewise, Professor. I'm gonna miss you.
okay, I'll miss you too. I'm going to, unless there's some other questions, I'm going to stop sharing and end the session. For everybody, goodbye.